So it's a real honor to be able to introduce Cynthia Keyon as today's Storer Lectureship speaker. Cynthia comes to us from UC San Francisco, where she has been since 1986. She is currently the Distinguished Professor of Biochemistry and Biophysics and American Cancer Society professor there. She also is the director of the UCSF Hillblom Center for the Study of the Biology of Aging. Cynthia did her graduate work at MIT in Graham Walker's lab, where she studied the induction of DNA damage repair genes. As a postdoc, she worked with Nobel laureate Sidney Brenner, and it was there that she began working on the nematode worm C. elegans. Her early studies on the worm helped to show that there was a conserved mechanism which governed the body patterning of all plants. All plants, right. You can see what organism I work on now. There was a conserved mechanism conserved mechanism for patterning the body plan of all animals. And it was actually, I was a graduate student with Cynthia, and it was that project that I worked on when I was in her lab. More recently, starting in about 1993, she started studying the lifespan, the regulation of the lifespan of animals. And it's this work, of course, that she'll talk to us about today. And one of the things that has characterized all of Cynthia's career is remarkable insight into the biology of the problems that she is studying. And with regards to the study of the regulation of lifespan, she really had a radical idea when she decided to start studying this. At the time, it was pretty much assumed that organisms aged because of the accumulation of damage, and there was no genetic program that regulated how long the lifespan of an organism was. And Cynthia, showing characteristic insight, thought that this was probably not true, and that actually there was a genetic program that would regulate the lifespan of organisms. And she set off to try and find genes that would regulate the lifespan of organisms. And I think that many people, including those of us in her lab, thought that this wasn't going to work, that it wasn't a good idea, and she proved us wrong, of course. And she showed that there was actually a single gene which could extend the lifespan of the worm by twofold. And even with this result, I think that many people thought, oh, that's just some funny worm thing. And Cynthia argued that that was not going to be the case, and has gone on to show that it is indeed not the case, that this same system regulates the lifespan of other organisms as well. So as I said, Cynthia has remarkable insight into biological problems, and I'll maybe talk a little bit more about that tomorrow when I introduce her for the scientific talk. But at this point, I think we should just welcome Cynthia and look forward to her lecture on lifespan regulation.
Thank you, Julian. It's really a huge honor to be here, and especially a great pleasure to be introduced by my former graduate student, who is a great star in our lab, and a great delight to have there. And I'm sure you all appreciate him as much as we do. So I'm going to be talking about aging, and you can see, now let's see, I think I have a pointer here, yeah, okay. So as Julian said, we all know what aging is, and we prefer not to think about it maybe, but there it is. And it is true that for many years, for centuries, I guess, people have thought that that aging is something that just happens. It's a completely passive, entropic, haphazard process. And then there's nothing that can be done about it and so forth. And I did start thinking that maybe that was wrong. And one reason is because if you look around in nature, you see different animals have really different lifespans. Here's some examples. A mouse has a 2-year lifespan, a canary 15, and a bat 50. My favorite is that a rat lives 3 years and a squirrel can live 25 years. So these animals, they differ just by their genes. Otherwise, they would have, anyway, that's what makes them have different lifespans. So that tells you right off the bat that genes have a huge effect on lifespan. And so that's the first thing. The second thing is that if you look around in biology, pretty much nothing happens just by chance. Everything seems to be subject to some kind of control. And so it seemed to me that maybe, like developmental pattern formation, which Julie mentioned, maybe there was some kind of regulatory control system for aging. And maybe, you could get in evolution these very different lifespans by changing regulatory genes. And it's a radical idea for thinking about aging, but it's absolutely mainstream when you think about pattern formation, because that really is how patterns evolve, largely by changing regulatory genes. So anyway, so I thought, well, maybe we could, if there are genes for aging, then if you change them, maybe you could cause the animals to live long. And if you could turn that around and you could say, well, if you look for long-lived mutants and you find them, first of all, you might not find them, and then you can just go home. But if you find them, then of course you can find out what they do by cloning the genes and figure out whether there is some kind of interesting control system. So now at the time, first I have to tell you that we didn't study this in humans. Instead, we studied it in this magnificent little creature, C. elegans. Here. This is an old individual here. And yes, okay, so now a lot of you know all about C. elegans. But I'll tell you, so basically it's a little tiny nematode, a roundworm. It's not an earthworm, it's a roundworm. It's microscopic, and it lives in the soil. It only has 1,000 cells, and it's a very simple, but it has lots of tissues that we have, like muscles and nerve cells and things like that. So it has a very short lifespan of just a couple weeks. So it seemed perfect for an animal to, look for mutations that affect aging. And but then, of course, you worry that maybe you would just learn about the worm. So the question is, could little C. elegans teach us anything about longevity in other organisms? Maybe lead us to the fountain of youth, for example, potentially. So I thought that might be possible. First of all, because as you know, probably ad nauseum by now, many biological mechanisms have been conserved to enormous extent during evolution. So, for example, the way muscle cells are made, the way the proteins work, the neurotransmitters are all the same. The way the cell cycle works, even the way patterns form in biology, they're all dictated by universal mechanisms that operate in worms and even yeast all the way up to man. So that was good because it suggests that if there were genes that controlled aging in the worm, then they might not be conserved, but then they would be an exception to the rule. And they're really easy to study, and as I say, they have short lifespans and they get old, as I'll show you in a minute with a little movie. Okay, so I was extremely enthusiastic to try to look for long-lived mutants. And at the time, there was already known to be a long-lived mutant that had been isolated by Michael Klass about 15 years earlier and studied by Tom Johnson. So I was very optimistic. It lived about 30 to 50% longer than normal. So, okay, so then I started asking rotation students if they wanted to work on this project, and they said no. And I actually have to say I tried to keep them away from the lab until they had committed to the project because the lab members thought I was totally crazy. Anyway, but finally we found one, a rotation student that is, that I shouldn't say it that way. But a very talented rotation student named Ramon Tabchiang, who's kind of a pioneer. And so he agreed to try to find long-lived mutants. And he did, and we were extremely lucky to find a mutation that made the animals live long. And so what Ramon found was that mutations that reduced the activity of a gene called DAF2 doubled the worm's lifespan. So here's a lifespan plot here. Here we have the age, and here we have the percentage of animals that are alive. So at time 0, the starting point, they're all alive. And as time goes along, they die. And what you can see is that the normal worms are all dead by the end of a month, whereas these long-lived ones are almost all alive at that time, and it takes them a lot longer to die. So they live more than twice as long as normal. But the really amazing thing about these worms is that they don't just check into the nursing home and hang on. They actually age more slowly. So this is a movie of a normal worm when it's a young adult, a graduate student. And here we have just 2 weeks later, brace yourself, this one's already dead, and this one is in the nursing home. See, its head is moving, but that's about all. So that when I say they get old, that's what I mean. And if you look at the tissues, you see that they look like they're deteriorating and they have age pigments and so forth. They really do get old. Okay, but at this time, our marvelous miracle worm looks like this. It's still moving around. Look, it's not quite as young as the graduate student. Maybe it's an associate professor, perhaps. Hoping to get tenure, for example. Anyway, so anyway, if you look at these animals with a high power microscope, what you see is that the cells look young at this time. So it's really slowing down aging. I mean, this is like looking at an 80-year-old person and thinking that they're 40. That's what this is like. It really is. So of course we want to know. I mean, it really, every time I look at it, I just can't believe it. So it's true. So we really want to know everything we possibly can about how you can change one gene and produce this amazing phenotype. Okay, so now the gene DAF2 was already known when we discovered its effect on aging. It was known because it affected an aspect of the life cycle of C. elegans. And I'm going to tell you what that is right now. So first I have to give you a little background. C. elegans starts off as an egg, and then it hatches. And if there's a lot of food around, it grows up through these different larval stages here to become an adult. But if it's in a harsh environment, by that I mean if there's a lot of if the worms are crowded together and the food is limiting, what happens is that they stop, they deviate from this growth pathway here, and they enter a state of sort of suspended animation called the dower. A dower would be analogous to hibernation or a spore, something like that. These animals, their growth is arrested, they don't reproduce, they don't eat, they're resistant to all sorts of things that you throw at them like high temperature or hydrogen peroxide, and they're very long-lived. So they just wait, basically. And then if you give them food again, they exit from the dower state and become normal L4 larvae, and then grow to be adults, and then they have actually a normal lifespan, even though they took this time out during the dower phase. So the dower is, you can consider it kind of a checkpoint. In other words, the animals go through puberty when they're L4s. So basically what's happening is the worms are saying, okay, well, should I go ahead and go through puberty and grow up and start reproducing or not? And if conditions aren't good, they wait. And if they are, they go through puberty. Now, once they pass this state, the L4, they can't go back. They can't become dowers. These animals, if they're hungry, they're just like a hungry person. They don't go into a dower state. Okay, so now the DAF2 gene controls this decision of whether to grow up to become an adult or to become a dower. If you turn the DAF2 gene off completely, what happens is worms hatch from eggs, and they always go into this dower state. They never grow up. They just arrest here. So the mutations that I was talking about that extended the lifespan with the movies I just showed you, those were mutations that inhibited the gene activity a little bit, but not too much. So it was still partially active. And so in other words, it had enough activity so that the animals could bypass this checkpoint here. They could grow up and become adults, and then they live long. Okay, so this was interesting. So the gene has two functions. It controls this dower decision, and it also affects the aging of the adult. So that made us wonder, what was the relationship between the dower and this long-lived adult? Were these adults just dowers in disguise? They're not. As I'll show you, they can be fully fertile, whereas dowers never have progeny. They eat, they move around, dowers are kind of thin and they look normal. So they don't look like dowers. They're not dowers. But the other thing we were wondering is, when in the life of the animal does the DAF2 gene act to control aging? Does it act at the same time early in development that it has to act to control the dower? Or does it maybe act separately in the adult to control aging? So it turns out you can ask questions like that very easily when you study C. elegans. You can use a method called RNAi, for those of you who know what that is. But basically, don't worry about it. What it is, it's a method that allows you to turn down the activity of any gene that you want at will. And you can also turn the gene activity down all through life or just during adulthood. So we did that. We asked, when does the DAF2... Oh, wait. Now, wait a minute. Hold your horses here. I'm going to go a little further. I'm going to first tell you what this gene is. Because, okay, sorry to jump ahead like this. I screwed up my slide. The DAF2 gene encodes a hormone receptor. It was cloned in the lab of Gary Ruvkin at Harvard. So here's a cell, and here's the receptor. It's like a baseball glove going through the membrane of the cell, grabbing the hormones as they come by. Okay, so now, this tells us a lot about what's happening during the aging process. the normal function of the DAF2 receptor is to speed up aging, to promote the aging process. Because when we inhibit it, the worms live longer. Okay. So this was a very important finding, the fact that DAF2 encodes a hormone receptor, because it demonstrates that hormones control aging. Okay, so now that I've told you that, I'm going to go back and tell you when in the life of the animal this hormone receptor acts to control aging. So we're going to go back a little ways. Here we are. Okay, so when does it act? Okay, so what we can do is we can turn the gene activity down at different times in the life of the animal. If we turn the gene down throughout the life, we live long, and that's what you would expect, because that's what happens when you have a mutation that changes the structure of the gene. Now what happens if you just turn the gene down only in the adult? Well, they still live long. So that tells you that the gene activity has to be normal in the adult in order for the adult to have a normal aging process. So in other words, the DAF2 gene acts during adulthood to affect lifespan. And we also did other experiments where we turned the DAF2 gene down during development and then back up during adulthood. So it was on in the adult and off during development. And those animals aged normally. So that told us that DAF2 acts exclusively during adulthood to affect aging. Okay, so if we go back to this life cycle here, what we know now is that the DAF2 gene acts early in the animal's life to control the Dower decision, but it acts again separately in the adult to control aging. So the aging process is really separate from this Dower formation process. Okay, so I've told you that the gene encodes a hormone receptor. So basically what this means then is that in the normal worm, there are hormones that speed up aging, which is a bizarre thought. You would think that you wouldn't think that the animal would have within it a kind of a grim reaper that's killing it, but that's what we have, is slowly killing the worm, the DAF2 receptor, and the hormones. Now, it was very interesting that DAF2 encoded a hormone receptor and that hormones control aging. It was even more interesting, in a way, that this hormone receptor was a very familiar one. It was very similar to two human hormone receptors, the receptors for the hormone insulin and the hormone IGF-1. And these are very well-known hormones. They're both essential. Insulin is known to be involved in food uptake, or the uptake of glucose, for example, into your cells after a meal. And IGF-1 is involved in growth. It promotes the growth of an animal. So our findings that in C. elegans, these hormones control aging raise the possibility that humans and other animals, this receptor might have a third role, which would be to control aging. And I'll come back to that in a little while, but immediately the sequence homology suggested that, the possibility. Okay, so now how does it all work? Well, we found a second gene that affected lifespan that turned out to be a very important gene. We found out that the longevity of these DAF2 mutants required a gene called DAF16. So here we have the long-lived DAF2 mutant, and in green, you see the lifespan of an animal that has the DAF2 mutation. So we've damaged the DAF2 gene, but we've also completely broken or damaged the DAF16 gene. And now they don't live long anymore. In fact, they have a slightly short lifespan. So we cloned DAF16 to find out what its function was, and it was also cloned in the lab of Gary Ruvkin. And it turns out that it encodes what's called a FOXO transcription factor. Now, what that means is that the normal function of the DAF16 gene is to make a protein called the DAF16 protein that goes into the nucleus where the genes are and turns other genes on and off. So DAF16 is a classic regulatory protein. So I just want to pause here for a minute and say that at the outset, I wanted to study aging because I thought aging was subject to some kind of control. And I think the fact that we found a hormone receptor and a transcription factor, a gene product that turns other genes on and off, says without a shadow of a doubt that aging is subject to control because these are control genes. These are regulatory genes. Okay, so now we have two genes. We have DAF2 and DAF16, a receptor and a transcription factor. So how do they interact to control aging? Well, first of all, this is a picture of what we know now it's a slightly simplified version of what we know now about what happens inside the cell when the hormone for DAF2 receptor binds to the DAF2 receptor. What happens is that a train of events is activated inside the cell. For those of you who are biologists, the DAF2 receptor activates a highly conserved PI3 kinase PDK-AKT phosphorylation cascade. There's just a set of proteins that are turned on, whose activities are stimulated by the binding of the hormone to the receptor. So what these proteins do is to inactivate this DAF16 gene by phosphorylating it. These little green dots here are phosphate residues. So when you activate the receptor, the DAF16 transcription factor gets these phosphate groups attached to it, and that turns it off. It makes it inactive. In particular, it keeps it from going into the nucleus, where the genes are, so it can't regulate the genes. If we inhibit the activity of either DAF2, as I've told you, or in fact these other genes here, then DAF16 can go into the nucleus where it leads somehow to lifespan extension. And I want to mention that our understanding of this signal transduction pathway here comes largely from the work of Gary Ruvkin's lab, but also with contributions from Jim Thomas and Don Riddle and Tom Johnson in our lab. Okay, so once we had shown that, my lab showed that DAF16's nuclear localization is regulated by the activity of this pathway. But then the big question became, what is DAF16 doing in the nucleus to double the lifespan of the animal? That's the really interesting question. So in a way, we were really lucky that DAF16 turned out to be a gene regulator protein or a transcription factor, because there are techniques now known as microarray technology or techniques that allow you to actually look at all the genes in the animal and ask which ones are more active or less active under some particular condition. So what you can do is you can ask, in our case, in a DAF2 mutant, which lives twice as long, if you look at what genes it's expressing when it's a young adult, is it expressing genes that are different from the normal worm? And it must be, because DAF16 is needed for this change in lifespan. So we did that. We looked for genes that were up or down in a DAF2 mutant, and in particular, genes whose change activity required this transcription factor. And sure enough, we found that DAF16 and DAF2 control the expression of many downstream genes. So again, when you turn down DAF2's activity, you increase the activity of this transcription factor, and then we found some genes get switched up and some get switched down, and there were a lot, maybe 100. I mean, there are 100 that were changed in a very substantial way. Okay, so that's great, but then the real question is, two questions. What are the genes, and do their activities really contribute to the lifespan of the animal? I mean, you could imagine that DAF16 could control 100 genes, but only one really affects lifespan. The others might do something else. So the first thing we wanted to know is, are all these genes contributing to the changes in lifespan that we see? And the answer was a surprising number of them were. What we found was that when we took a DAF2 mutant, the long-lived DAF2 animal, and we knocked down the activity of any one of these downstream genes, we found that they didn't live as long. That wasn't the case with all genes, but with many genes it was the case. So here you have another lifespan plot here. Here you have the long-lived DAF2 mutant in black. And if we turn, for example, for comparison, the DAF16 transcription factor off using this RNAi method, they have a short lifespan. But if we turn any one of these particular downstream genes off, they have a shorter lifespan. They don't live as short as DAF16, but they don't live as long as DAF2 mutants. So these are genes whose increased expression is contributing to the long lifespan of the DAF2 mutant. And I told you that some genes were actually turned down in the long-lived animals. So it was interesting to ask, what would happen if you turn these genes down in normal worms? Would that make the normal worms live longer? And it did for many of them. So many of the genes that were turned down in the DAF2 mutants also influence longevity. So in this case, here are the normal wild-type animals. And when we turned down these genes that were normally turned down in the long-lived animals, we increased lifespan. Okay? So that said, that there were classes of genes that affected lifespan, not just one gene, many genes, and it was what you expect, the ones that were turned up in the long-lived animals tended to contribute to the long lifespan. The ones that were turned down, it was good that they were turned down because they would have inhibited the long lifespan. Okay, so now the big question, what are they? Well, all the genes have been sequenced, so we just know instantly what they are. And it turns out that these genes do many different things, not just one. There's all sorts of theories for how you age or what causes aging, and so it was extremely interesting to find out what these genes actually do. So this is a list of the genes whose functions contributed to the long lifespan of the animal. There was a whole class of genes that encoded antioxidant proteins. So these included superoxide dismutase and metallothionein, glutathione transferase, catalase, and various others. Some of these had been found by other people, like Gordon Lithgow, to be regulated differently in the long-lived animals. And we found more of them, and we also found that their increased expression or activity was important for the long lifespan. So that's really great. I mean, this idea that free radical damage is generated during part of normal metabolism and that accelerates the aging of the organism is a very strongly rooted hypothesis in the aging field. And I think that our findings suggest that it's absolutely true, that the reactive oxygen species probably really do accelerate aging. We also found that some genes called chaperones, in particular for the scientists among you, the small heat shock proteins, were increased in activity in the long-lived animals, and that increased expression was important. Now, chaperones are proteins that take care of other proteins and look out for them, just as their name suggests. So if a protein can't fold correctly, a chaperone can help it to fold, or if a protein is really damaged badly, the chaperone can actually escort it to the garbage can of the cell so that you can make new proteins. Interestingly, we found a whole bunch of genes that were kind of, they were like the immune system of the worm. They were genes whose products kill microorganisms. And we actually had done experiments previously that showed that worms probably die from infections because they eat bacteria. So they're just living in a sea of bacteria. And it turns out if you prevent the bacteria from dividing with an antibiotic, then the worms live longer. Not as long as the DAF2 mutant, but maybe 30% longer than normal, suggesting that, and they also get packed completely full of bacteria normally. So I think having these antimicrobial genes expressed at higher levels or more active could help the animals to live longer. And actually, the Ossibel and Ruvkin labs found that DAF2 mutants are resistant to all sorts of bacterial pathogens, consistent with this finding here. Then there were a variety of metabolic genes that were whose expression was affected that were important. The most interesting of those, I think, were a set of genes whose normal function is to transport fat around the body. Now, these genes were turned down in the long-lived animals. And we know they're important for lifespan because when we turn them down in normal animals, the normal animals can live long. And we don't exactly know why fat transport is shortening the lifespan of the animal. We don't understand that. But what's really interesting is that the human versions of these genes seem to be responsible, at least in part, for the ability of some people to live to be 100. So let me phrase that differently. You know what a centenarian is. That's a person who lives to be 100. The ability to live to be 100 runs in families. And gene mappers have found genes that seem to be contributing toward this longevity. And there are 3 different ones. And all 3 of them seem to be involved in fat transport. And one of them is the human version of one of these genes here. And it actually has binding sites in front of it for DAF16. In other words, the DNA sequences upstream of it suggest that it could bind to FOXO family transcription factors. Whether it does or not, we don't know. But it's intriguing. Okay, so now I just want to talk about the big picture for a while here. So basically, what we find here is we have some control genes. We have a hierarchy. Here we have the sort of master regulatory genes here, control genes like the president, let's say. Well, I shouldn't use that example. But anyway, sorry. Okay, so we have controlling genes regulating many subordinate genes that do different things. This is a kind of a coordinate control circuitry. It's like an orchestra where we have the orchestra conductor, which would be DAF2 and DAF16, maybe 2 conductors in this case. And here we have the violins and the cellos and the French horns and the piccolos and the flutes, etc. The different instruments doing different things. That's the important thing, doing different things, but doing them all together in concert so that you get a big effect. And I'll remind you that on their own, each one of these genes, when you knock it down with RNAi, didn't produce a very big effect. It has a minor effect, suggesting that the dramatic change in lifespan comes from the cumulative activities of all these different genes acting together, doing different things. So there's lots of implications. The first is that in the genes of the worm, in the DNA, there is encoded a lifespan regulatory module. There it is. In other words, there are genes for DAF2, DAF16, and for these metabolic, antioxidant, and everything. And they're all wired so that they're linked together in the way that I showed you. So the ability for these worms to live a lot longer is really encoded in their DNA. We just had to tweak it the right way for it to express itself. Here's the regulatory module again. Okay, now how could you evolve something like this? Well, remember that the DAF2 gene, and also DAF16, by the way, controls dower formation. Now, the dower is, remember, it's very advantageous for an animal to be able to enter a stage like the dower stage. Let's suppose you have two populations of worms. One can become dowers, one can't, and food is running out. Okay, so our dower formers, they go into dower and they wait. Nothing happens. The other ones go up, try to have progeny, the progeny die. And then so you see, it's obvious that it's of incredible value to the animal to have evolved a system like that. Now, dowers also have increased expression of a lot of the different genes that I just mentioned that affect the lifespan of the adult. And these genes, like the antioxidant genes and the chaperones and so forth, allow the dower to be resistant to all those stresses I was talking about, to high temperature, to reactive oxygen species generators like hydrogen peroxide. So here's what I think. I think it's very likely that this lifespan regulatory circuit didn't evolve to regulate lifespan, although it may have. But it's very likely that whether it could have or not to affect lifespan of the animal, it certainly would have had a reason to evolve for the dower. But once you have it there, once it's in the genes, the regulators and the downstream genes, once they're physically there, then automatically you have a system that if it's running in the adult can influence the lifespan. And here's the reason, because I think the same genes that can keep an animal resistant to something like temperature or desiccation or something like that can also protect a grown-up animal, an adult, from aging. In other words, reactive oxygen species can be generated by the environment, by UV light, for example, ultraviolet sunlight, or they can be generated by your own mitochondria. So if you have the one and the same protein can, number one, make you live long, number two, make you resistant to that kind of stress. And in fact, Tom Kirkwood's lab did a really interesting experiment. They took cells, fibroblasts, skin cells, from lots of different kinds of mammals. And they asked, if you put these cells in culture and you dump hydrogen peroxide on them, how resistant are they? How much hydrogen peroxide does it take to kill them? And what they found was that animals that had longer lifespans in general tended to have fibroblasts that were more resistant to hydrogen peroxide. Okay? Again, suggesting that there is a real fundamental link between stress resistance of cells and the longevity of the animal from which it came. So I think this is really great. I mean, this is a way to finally explain how, because when I first thought about studying aging, the evolutionary biologists had all these theories for why there wouldn't be any, or not theories exactly. I mean, they thought it was true and settled and everything that you couldn't have regulatory genes for lifespan because aging happens after you've already reproduced. But this is a mechanism you see that could evolve to help an animal before it's reproduced. Okay. Yes, in the same. Okay, good. Furthermore, I think this provides a nice possible explanation for how you can generate these different species in different kinds of animals. I told you about the bat and the mouse and the so forth, but it turns out there are long and short-lived insects, long and short-lived birds, and like hummingbirds just live a few years, and I think parrots can live to be 100, and long and short-lived mammals. So there must have been a first bird and a first insect and a first mammal, and it must have had some lifespan, suggesting that changes in lifespan have evolved not just once but many times. So I think having a lifespan regulatory module like this suggests maybe that it's possible to make big changes in lifespan, maybe by changing the regulator genes, or perhaps by changing some of the downstream genes. Like, for example, if you made a mutation in a DAF16 protein that caused the protein to bind more tightly to the target, so it turned them on higher, for example. I don't know that that would be the case, but something like that could happen. Maybe that could have a big effect on lifespan all at once. So I also just want to mention that DAF16 is turning out to be a really important regulator. I told you that this is this transcription factor. I told you that it's controlled by these hormones, the insulin IGF-1-like hormones. It turns out that activating any of a number of different pathways, the heat shock pathway, the Jun kinase stress response pathway, the heterochronic gene pathway, or a gene called SIR2. Now, for the non-scientists among you, the point here is that these are just different pathways, all of which lead to stress resistance, but none of which have anything to do with insulin. Activating any of these can extend lifespan. And in all cases, you require this DAF16 gene. So this was from the work of these labs here. So I think that's really interesting because it suggests that there's not just one way that you can tweak the system to increase lifespan. I think there are many ways that you can do it. So now the big question, of course, is this just a worm thing? And the answer is no, it isn't. It turns out if you make the same changes in the insulin IGF-1 receptor in the fruit fly, flies live longer. And if you change, now mice have separate receptors for IGF-1 and for insulin. And if you change the IGF-1 receptor, normally a mouse has 2 copies of this gene, one from the mother and one from the father, just like all of the genes a mouse has. If you damage one of them or take one of them away, so it only has 1 copy, it makes half as much receptor for IGF-1. It turns out it lives longer. If you take the insulin receptor away from the fat tissue, the adipose tissue of a mouse, the whole mouse lives longer. These mice actually don't get fat if you feed them a high-fat diet. So they're very lucky mice. And these ones here are quite lucky too. I mean, they live longer. They have normal reproduction, normal metabolism. So it's really quite remarkable. It's also remarkable that it wasn't just one of the receptors, but both of the receptors that seem to be involved in lifespan control. I mean, it's just really remarkable. So now these changes aren't as large. So far, these are in the order of 30 to 40%, even less, maybe 20 to 40% for different kinds of mutations in these, whereas these can be as big as 80% here. Okay, so this is really, obviously, for us, I mean, it was thrilling, totally thrilling to discover something in C. elegans and have it be conserved, especially if it's aging and you're worried about getting old, although who knows, we don't really know yet for people. But we do know for animals. So actually that's something, because you could, I'm just going to skip that, because at least maybe there's something for animals. Okay, so what does it all mean? First of all, why would insulin and IGF-1 be influencing aging? I said that the normal function of these 2 hormones is to promote growth and food storage. And I think the situation is that an animal can be in one of 2 kinds of states, maybe many states, but at least these 2 states. One is a state of growth, which is accompanied by more rapid aging. But I think that low insulin IGF-1 signaling or activity of these other stress response pathways can shift the animal from a state of growth and activity into a state of maintenance, where you make proteins that protect the antioxidants, chaperones, things like that. And I think that's what we're doing, switching from one to the other here. And again, I think it's really cool that this network is written into the genes. So I think, and there's a lot of ways to get into it. So I think it's possible, perhaps, that someday we'll be able to increase the youthfulness and longevity of people this way. Who knows? It's not clear, but it's possible. Okay. Oops, I'm pressing the button. Now, the other thing I want to mention is that it's not just aging that's delayed in these long-lived animals. Also, the diseases of aging are delayed. This is just a list of what other people have found. These people's names are mentioned down here. In C. elegans, you can cause C. elegans to get Huntington's disease if you put the Huntington's disease gene into C. elegans. It's a protein aggregation disease. It's a disease of aging. And the long-lived worms, they get it, but not till later. They also get, normal worms when they age, get a muscle wasting condition that resembles sarcopenia in humans. That's postponed. Heart failure in Drosophila happens when a fly gets old. If you stress its heart, the heart fails. It turns out in these long-lived mutants, the heart never fails. Even if you stress it when it's on its deathbed, the heart it seems to be in better shape than the rest of the animal. So there's a real cardiovascular benefit here. And also, these long-lived mice are resistant to cancer and various other ailments. So it seems like there's a whole host of age-related diseases which are postponed, or else they don't even happen in these long-lived animals. So I think it's very important because it suggests a new therapeutic strategies for going after all sorts of age-related diseases all at once, namely by going after aging, by trying to target some of these pathways that I've been describing to you. Okay, so now I want to ask a few fun questions. The first question is it possible that some kind of environmental signal could affect this aging system? And obviously, the one that springs to mind immediately is caloric restriction. Because you probably all know this, but for rodents and yeast and worms and flies and maybe even primates, if you don't eat as much as you would like to eat, if you restrict calories, you live longer. And you don't get cancer, you don't get a whole variety of diseases. It's really quite an interesting phenomenon. Now, when you calorically restrict an animal, you lower the insulin level of the animal. So since insulin would activate the DAF2 receptor and speed up aging, it would make a lot of sense that this system would be engaged by caloric restriction. And it turns out, amazingly enough, in the worm that it's not. Because here's why I say that. If it were the case, this is what would happen. You would eat less, so you would have less insulin, so the DAF2 receptor would be empty, so activity levels would be lower for DAF2, so you would live longer. Oh, DAF16 would go up, the transcription factor would be turned on, and you would live longer. So if that's true, then in order for caloric restriction to extend lifespan, you ought to have that transcription factor. That should be necessary. But it's not. It turns out you can completely eliminate the DAF16 gene and calorically restrict the worms, and they live just as long. So in worms, this system is not mediating the response to caloric restriction. It may be in mice, actually. There's some unpublished evidence suggesting that may be the case in mice. But in the worm, it's not. So what we found, though, is that environmental signals probably do play an important role, but the worms don't seem to be eating them. They seem to be smelling them or tasting them. It looks like sensory perception may influence aging. And this is what the nose of a worm, the face of a worm looks like. It's beautiful here. Here are the nostrils, here, where the sensory neurons are. And these are the neurons that let the worms go towards things they like and away from things they don't like. And if you make a mutation that inhibits the sensory neuron activity, then worms live longer. All these are mutants with defective sensory neurons. And actually, we can cause the worms to live long if we take away a gene or knock out a gene that makes what's called a chemosensory receptor. So this is a protein that is present only in the very tip of the nostril, with one side is sticking out toward the environment. And these are the proteins that actually bind to the things that you smell or taste. And if we remove one or a couple of those, we can extend lifespan. The animals eat normally. They are not calorically restricted. So we think somehow that sensory perception or smelling and tasting is affecting their lifespan. So here's how we think it works. We think there's some kind of signal in the environment that can change the activity of the DAF2 insulin IGF-1 system in the animal. This is a nice hypothesis for several reasons. First of all, they do need DAF16 to live long. So if you change the sensory perception, you get long lifespan only if you have DAF16. So that's one thing. Also, DAF16 goes into all the nuclei in the animal, if you can watch that happen, even though you only change the tip of the nose. Also, these sensory neurons have been shown by others to express genes encoding insulin-like peptides. So we don't know for sure, but our model is that the activity of the sensory neurons, the normal activity, causes the animal to release the ligand, the hormone for DAF2. It binds to the receptor and speeds up aging. So when we come along and we interrupt this train of events with a mutation, or with a laser even, then we don't release so much hormone, so it's like having a DAF2 mutation, and you live longer. Okay, so then we've been starting to ask some interesting questions. Which neurons influence lifespan? And what we find is very interesting. We find that here's what the cell bodies are. These are where the nuclei are for all these neurons down in the brain of the worm. If we kill just one here, then the worms live long. So this is a neuron that affects lifespan. It turns out, interestingly, this is a gustatory neuron. That means it's a neuron that responds to soluble compounds, or it's a taste neuron. If we kill that same neuron, plus this neuron though, we don't increase lifespan, which is amazing. That tells us that not all these neurons are doing the same thing. Some neurons are promoting long life and some are inhibiting long life. So we have different kinds of neurons. In fact, we find that if you can also get the animals to live long if you kill olfactory neurons. These are the smell neurons, neurons that allow the animals to respond to volatile compounds. And it's interesting, I told you that killing this neuron can reverse the effect of killing that one, another taste one. If you kill this one here, which is this one here, which is a smell neuron, they live long. But killing this neuron does not reverse it. So killing this guy reverses the taste problem, but not the smell problem. So it looks like the taste neurons are having a little tea party by themselves, and there's another one going on for the smell neuron. So there's a whole bunch of complexity up here in the brain of the animal, which is going to be really interesting to figure out. So, okay, so these and other findings suggest to us that the lifespan of the worm is influenced by something in the environment. We don't know what it is. I suspect it's something that signals hard times are coming, but I don't know. We're doing things like growing worms on bacteria that have been traumatized in various ways. It turns out that it probably is going to be the case in flies also. There are unpublished suggestions that olfaction in flies also affects lifespan. And who knows about humans? I'll just tell you, though, if you're eating a meal, the level of insulin in your body goes up. And if you also smell the food that you're eating, it goes up even more. So who knows? Who knows? Okay, so I see that I'm out of time. I'm just going to jump ahead through these slides and tell you something pretty cool. This is about reproduction and aging, which I'm going to talk about in my specialized lecture tomorrow. But let's just ask the question, how long can a worm live? So what I haven't told you is that the reproductive system also makes signals that affect aging. And so now I'm going to tell you that when we alter the DAF2 gene, plus the reproductive system in the same animal, we do two things in the same animal, now they can live 6 times as long as normal, which is amazing. So this is the normal lifespan, 20 days, and these guys live 126 days. So, and the thing that's really just unbelievable is that they're really healthy. These are worms that were photographed on day 144, and they are moving around. So remember at the movie I showed you earlier, I showed you some worms at day 13 or 11, I forget, 13 I think. And one was dead and the other one was on its deathbed. But these guys are actually quite happy. In fact, Nuno Arantes Oliveira, who is a graduate student studying this, took the petri dish with these worms on it around to other lab members and said, how old do you think these worms are? And they said, oh, maybe 5 days old. But they're 144 days old. So I guess I'm just showing you that because it really makes you wonder just what's possible. So I'm going to spend just a little bit of time talking about people, just because this is a public lecture. Here are two. This is not my joke. This is from Colleen Murphy, my former postdoc. Could we stay young longer? Okay, so it's fun to think about this. And first, I just want to start off by saying maybe not. Maybe there's something, we've reached our maximum, there's nothing we can do about it. That's possible. But I just want to remind you that even though I just told you that we can increase lifespan of worms by 6-fold, it's nothing compared to what evolution has done. Because evolution, going from a worm lifespan to our lifespan, just by changing genes, has increased lifespan more than 1,000-fold. So that's really a lot. Okay, the other thing, next thing is the germ lineage is immortal. And I know this is going to sound like I'm not saying anything, but immortality is a really long time. I mean, forever is a long time. It's a lot longer than 100 years or 500 years or 1,000 or 10,000. It's forever. So I think that's just, it means it's possible for at least for a cell lineage to live a very long time. Now, you can think about aging in the following way. You can think about it being kind of a tug of war, if you will, between damage, which is propelling you toward old age, and prevention and repair, which are opposing that. So you can imagine that what determines lifespan are the relative rates of these two processes. So for example, a mouse ages quickly because the rate of damage is pretty high. Right, let's see. Let me go back here. Okay, so let's start with a long-lived animal. So in a case of a bat, for example, which has a long lifespan, you would imagine that the ratio of prevention and repair to damage is higher than it is in a mouse. Same with a squirrel versus a rat. Okay, so if that's true, then you could just imagine like a thought experiment of boosting the prevention and repair ability of a cell until it equalizes the force of damage. And if that were the case, if that could be done, then it wouldn't age, potentially. Okay, so now you ask, well, all right, could we live longer? The first question is, why do we live as long as we do? We have an incredibly long lifespan. We probably live so long for two reasons. All animals have a strategy for living. The worm's strategy is to turn whatever it sees into new worms as soon as possible. It has a 3-day generation time, and it produces 300 progeny. But we don't have that strategy. That's not our, fortunately, in my opinion. Anyway, our strategy is to control resources, the environment, and to organize into groups that can do things. And I think we live so long partly because it's useful for a tribe to have a few wise elders, people who would remember what to do. You know, if a plague came through 50 years ago, they might remember what to do. And also possibly for the grandparents to help take care of the grandchildren. Okay. And actually, if you look at, it's interesting. You might think, why do we listen? But if you look at who's running the world, who are the CEOs of the major companies, who are the leaders of the major countries of the world, politically, economically, financially, who are they? Most of them are not people in their teens. They're plenty old enough to have children, no problem. But you see what I'm trying to say? These are people who tend to be grandparents, and not always, but in many cases, which just tells you right off the bat that there must be something of value to age in the human race. Otherwise, the 20-year-olds would be running all these companies. They're stronger, after all. Okay, now why don't we live longer already? Well, I don't think there's a lot of selective value, perhaps, in living longer. I mean, if you already have the grandparents around, do you need the great-grandparents? Maybe not. And maybe things don't come. You see what I'm saying? So maybe we have the lifespan we do because it's optimal for the way we live. And maybe there isn't a barrier for us potentially to having a longer lifespan. There might be, but there might not be. So I just want to, also, people have never really taken this seriously because we don't have a role model. There's not some primate that can go to the opera and do arithmetic and so forth that lives to be 300. It isn't. If there were, I think, then we would already have been studying this for a long time. For example, I think we invented invented airplanes because we could see birds fly. So they were our role models. So I think maybe these long-lived mutants can serve as our role models now. And I'll just end by talking about the people who did the work. Ramon was this fearless rotation student who found that DAF2 mutants live long and started all this off. Andy did the timing experiments I told you about. Quay cloned the DAF16 gene and showed that it was a transcriptional regulator. Colleen did the microarray work. She discovered that there was this longevity network. Javier and Joy did the work on sensory perception, and I didn't talk about the work of Douglas and Honor on the reproductive system, but Nuno was the one who found that he could make worms live 6 times as long as normal and stay healthy. So thank you. I'm sorry that I went over the time.
We have time for some questions.
Okay.
It's hard not to listen to this and wonder about costs.
Wonder about costs?
Wonder about the costs of greater longevity. And you mentioned one, and that is the likelihood that, the knowledge that these worms are less able to respond to stress by forming this resistant state. Which I can't remember.
No, I'm sorry. The long-lived animals are much more resistant to stress.
But they can't go into that separate pathway, life history pathway, can they?
Oh yeah, they're actually potentiated toward it. They go into it more easily, which in a way could be a problem.
So then if there's no cost there, why aren't natural C. elegans populations completely dominated by alleles for these regulatory genes that allow them to live long?
Well, because I think C. elegans, like I was saying, it has this strategy of turning anything it sees into worms as soon as possible. So it's like a bank account that has a 300% interest every 3 days. It's really fast. You know what? So what happens is you start with one worm and then some roadkill, bacteria, and you put a worm there. So the worm eats all the bacteria, and pretty soon it's just worms. In fact, we did that. We took eggs and we put a little hole and put, Steve Salser did that, and put a worm into the egg, and pretty soon you open it up and it's nothing but worms. So that, and very quickly. So if you think about it, after 6 days, the first worm, the egg that you put in there, has grown up and has had 300 progeny. So now you have 300 brand new adult worms and one 6-day-old worm. So what's the point, really, of putting your resources into a worm that why keep it alive for a really long time? If you have already made brand new worms, it may be just cheaper to throw it away and use the new ones. So I think that could have something to do with it. Yes?
So along these same lines, can you, I always think of trade-offs too. Can you produce a worm that lives 60 days, reproduces normally, and stays active for 50 out of those 60 days?
The best we've done, I think, now these worms that were living 6 times as long, we changed the reproductive system in such a way that they don't have progeny, so they don't count for your question. Okay. The reproductive system seems to stimulate the production of a steroid hormone, and if we could do that some other way, I think they would be fertile, but we don't know how to do that yet. In the case of DAF2 mutants, if we take normal worms and we let them grow to become adults, and then we put them on DAF2 RNA, we turn down the DAF2 gene just in the adult only, then they live twice as long as normal, and they have normal reproduction. And they're very healthy. Now, I have to say, though, they're very active.
All the way to till they die?
Well, not all the way. It's as if it takes the long-lived worms 2 days to age as much as a normal worm ages in 1 day. So it has a longer period of activity, but it also spends a longer time in the nursing home. Just like an old dog is old for a less absolute value of time than an old person is, let's say, elderly, I would say. On the other hand, those mice, when you change the insulin when you take away the insulin receptor from the fat tissue of a mouse, it's only been done once in one experiment, but it was very interesting because what happened was when all the normal mice were dead, almost all of the altered mice were fine. They were moving around, they were just great. And then they died, but they died with the same kinetics as the normal ones. So it was if they had just expanded the youthful period and then had, but not the old age period. So it seems like it sort of depends on exactly what you do. Yes. Yes.
Does either DAF2 or DAF3 have an effect on chromosome N?
Chromosome what?
N.
Oh, telomeres? Probably not. No, the DAF2 mutants have, so, okay, I have to describe that to you. There's, the chromosome ends are called telomeres, and they can get shorter when cells replicate if there isn't a particular protein called telomerase around that lets them get longer. And people, for example, after they're young, the level of this telomere building protein goes way down. So as our cells divide, the telomeres seem to get shorter and shorter. So the question is, and people found that you could keep cells in culture alive, maybe forever, by giving them high levels of this telomere building protein, telomerase. So the question, that you probably all read about it in the newspapers, the idea was that your telomeres were kind of the fountain of youth. Or if you could fix them, you could remain alive forever and so forth. It looks like most likely telomere length doesn't have anything to do with this. These long-lived animals have normal telomeres. And if you just let worms grow just naturally and you just take populations of them, if you just put one worm on each of a number of petri dishes and get big populations out of each one, and after a while you measure telomere lengths, they've randomly changed. They're not all the same, but the length of the telomeres doesn't correlate with the lifespan of the animal, suggesting probably that it doesn't matter in the worm. Which isn't to say that it doesn't matter in humans, and it may actually matter in humans.
With regard to the oxidant component of aging that you mentioned is involved, there's a group at Berkeley that believes mitochondrial oxidative effects are modulated by alpha-lipoic acid. Have you looked at that in any of your model, or do you have?
No, we haven't done that. That's Bruce Amos's work. No, we have never done that. We've talked to Bruce about doing that, and we never have. But if you give, I think, rats alpha-lipoic acid, he says they start dancing. And so they're very healthy and they have better mental acuity, but they don't live longer, apparently. So, but it's good. I mean, it's fine. Oh, well, this person has also wanted to.
We'll do two
We should have
questions.
two. Okay.
So the effect of ablating sensory neurons on lifespan, is that specific to certain types of chemosensory neurons? Or
Well, we just started, Joy Alcedo with the postdoc, she just started to kill individual ones. So the first thing we did is, we can take away the nostril for those noses, and then we find that that alone can produce an effect. So we know the neurons in those nostrils are important, and there's 12 in each nostril. And so you can't really kill all combinations. It's so hard. You have to kill, it's really a hard thing, because even to do one lifespan, you have to kill about 80 animals. It's very difficult, one single neuron. So she did just a few, and what she found was, like I said, that there were some neurons that respond, so there are neurons that are known to respond to be taste neurons and smell neurons, and either one or the other could have an effect. If she killed them both, they lived even longer. And then some neurons are known to respond to some foods that the worms eat. But if she killed those, or at least this one in particular, there was no change in lifespan, which is why I think it's not just everything. It's something specific that the animals care about, some combination of things that they smell and taste.
so how about thermosensory and mechanosensory neurons?
We don't think mechanosensory ones are important because we looked at the lifespan in one of those. And we've also looked at the lifespan in some thermotaxis mutants, and there's at least some of them that seem to have a normal lifespan. Not all, but then on the other hand, they're not all specific. So some of the ones that don't also affect other neurons as well.
I might have missed it. Do you actually have also mutants which are making the deaf inactive only very late in life? So you have, let's say, your 14-day-old wild type, and suddenly
Yes,
deaf
yes.
goes down, and is it then going down, or it continues its life?
We don't know. We haven't got a way of doing that experiment properly. The way we do it with RNAi is we can turn down, we can cause the mRNAs to go away. But if the proteins become more stable later, so what we find is that up through reproduction, like when the worm is halfway through its life, if we add the RNAi treatment at that time, we can still get an effect. But we don't know. And there may be a change in protein turnover with aging. It looks like there probably is. So it would be nice to really do that experiment. Yeah.
Please join me in thanking Cynthia.
I got a little tongue-tied.